Welcome back, everybody. This is lecture number 24. We're going to talk about unusual infectious agents. I haven't given this lecture since 2017, but this is really interesting because, as the title says, they're unusual and different. So I'm really excited to do this again this year. And if there's anything that connects all the things we're going to talk about today, it's this. I don't think you have to connect it all, but if you had to, it answers this question. What's the minimum genome size needed to sustain an infectious agent? Or maybe even more interestingly, could an infectious agent exist without any genome at all? And what we'll talk about today are viroids, satellites, and prions. And they're going to answer these questions. That's a viroid, that's a satellite, and that's a prion. I mean, just the words are great. I love viroid, right? Satellite's okay. Prion is just and that's an invented word. So let's start with viroids. These are just amazing. Circular single-stranded RNAs. They range in size from 120 to 475 nucleotides. And here's a diagram of a viroid right here on the right. The green is the RNA. And the RNA is extensively base paired, as you can see here. So it looks like a rod under the electron microscope. So it's mostly base paired. There are loops, as you can see. And this doesn't code for any protein. There are no protein coding regions. And there's no protective coat. There's no capsid. There's no nucleocapsid. There's no envelope. It's naked RNA. And yet it can go from host to host. The host of viroids are plants. You don't need receptors. And these get into plant cells. They can get it in a variety of ways by mechanical damage. Farmers often introduce them when they run over plants. They can be introduced by vectors, insect vectors, worms. And when they get into the plant, they replicate. And so that's why they were originally discovered. They can do things to plants. And T.O. Diener, who discovered them, called them viroids because he said they're not viruses. So let's get another name, and viroid it was, and I think this is a great name. There are two families of these viroids. There are the PSTVd. So you know the virus families are viridae, so this is viroidae. Those replicate in the nucleus of the cell, and the avsun viroidae replicate in the chloroplasts. And these are not random names. These names are named after the disease caused by the viroid. Avsun stands for avocado sunblotch viroid. How about that? Isn't that just the best? Now, some viroids are ribozymes. I don't think we've talked about ribozymes in this course, but these are enzymes based entirely on RNA. And so within this RNA sequence is a sequence which basically has self-cleaving activity. And for those viroids that have ribozymes, these activities are essential for reproduction of the viroid. That activity without a protein is probably a relic of the RNA world. These probably existed way back when. You know, in the RNA world, RNA was the catalyst. It did everything. And so this is kind of a relic of that. Now, I like to make this distinction from the virus quote, lifestyle. Viruses were parasites of the translational machinery, right? According to the Baltimore scheme, every virus has to make mRNA that can be translated by the host cell. So they're parasites of translation. But viroids are parasites of the transcription machinery. They're not translated, so they're not parasites of that, but they need to be copied by the transcriptional machinery, as you'll see in a few moments. The first viroid discovered was potato spindle tuber viroid, PSTVd, discovered in 1967. Of course, they've been around for a long time. We just found them because during the production of potato seed stocks. So to this day, potato seed stocks have to be produced that are viroid and virus-free. And a couple of years ago, I went up to Cornell in Ithaca to visit, and a plant virologist showed me the place where he makes potato seed stocks free of viroids and viruses. And he supplies the farmers up in upstate New York. with them, because eventually the farmers will plant them and they will make crops, and then year after year they will plant them, and then at some point they get infected and they go back to the virologist at Cornell and get new virus-free seed stocks. Very cool. This one was 359 nucleotides. So it's the first discovered, and since then we've discovered many more. And some of these are what I say is benign. They don't do anything to the plant. But others can cause diseases of crop plants that are economically important, and that's why they were first discovered. The potato viroid makes alterations in the potato plant. And you can see an example of that here. This is a tomato plant. Here's a healthy plant without any viroids. And then these are tomato plants that had been infected with 3 different viroids. And you can see they have different effects on the growth of the tomato plant. And so you don't want these, if you're growing tomatoes for a living, you don't want these in your tomatoes. And some of my favorite ones I have to tell you before we go on. The Kadang coconut viroid, CCCVD, causes lethal disease of coconut palms. And if you like pina coladas, you're sad. This is what it looks like. You ever see a palm tree? You go somewhere where they have palm trees. If they're brown like this, that's Kadang coconut viroid. Lethal disease. It will kill the coconut palm. Now, on the other hand, the hop latent viroid is no symptoms in the hop plant. And of course, beer lovers are relieved because these plants are fine, even though they have this viroid. And finally, the apple scar skin viroid makes apples look like this. The apples are fine. They taste great, but people don't want to buy them. If you saw this in a supermarket, you probably wouldn't buy it, right? But if you do in the future, now you know it's okay. It's just a viroid, and it's not going to hurt you, of course. It'll just pass through you. Someone should do something with Apple computers in this, right? There's got to be a joke in there. Now, viroids have functional regions. Even though they don't code for proteins, different parts of the RNA has different functions. You know, there are 2 ends. So this is potato spindle, a member of the PSTVd. You have the left and right ends. Then you have a region here called pathogenesis, and we're going to talk about what that does in a moment. It is involved in making the plant sick, and if you change it, you can reduce the pathogenesis. There's a central conserved region. There's a variable region as well. And then at the bottom here is a member of the Afsun viroidae, but this one has the ribozyme. So you can see these are longer and more extensively base paired. This sequence right in here is the self-cleaving sequence, the ribozyme, and it will cleave itself. And the reason for that will become evident to you in a moment. You know, why these things should have that. How are these transmitted? There are both vertical and horizontal transmission. In vertical transmission, here on the left, the infected pollen, so the pollen contains the viroid, contacts the stigma, so that's the normal process, right? And the viroid moves through the pollen tube, that's the red line, all the way to the ovule, from where the seed is going to be produced. And then the viroid ends up in the seed, and when the seed germinates and becomes a plant, then the seedling is infected. So that's vertical transmission. You know, like mother to child. And then we have horizontal transmission here. We again have infected pollen from an infected plant. It simply transmits it to the other plant the same way, except that it's not necessarily transmitted in the seedling, although in this case it probably is, but it can get into the other plant tissues as well from that infected pollen. So 2 different ways of transmission. So you can see the pollen, of course, can be moved by bees, right? Normal process of pollination, or just some kind of mechanical transmission as well. Now, how do viroids replicate if they don't code for any protein? They're copied by host RNA polymerase II. And so this is the exception to the rule I told you way back in the beginning, that viral RNAs can't be copied by cellular enzymes. This is the exception. The viroids can be. RNA polymerase II, the enzyme that normally copies DNA to make mRNAs, right? It will copy these. I'll show you the details in a moment. The RNA polymerase makes concatamers of these viroids, and you know now what a concatemer is. And in one group of viroids, the ones with the ribozyme, the name for it is hammerhead ribozyme, it cleaves the concatemers. And so here's the formal definition of the ribozyme: an autocatalytic self-cleaving RNA. This ability of RNA to make an enzymatic reaction without protein was discovered in 1981 by Tom Cech at the University of Colorado, got the Nobel Prize for that. Ribozyme is used to cleave the concatemers during replication. Viroids in general make concatemers, and the ones that don't have a ribozyme, they're cleaved by nuclear enzymes. Someone asked if the plant viroids cause disease in insects. No, they don't. And you'll see why in a moment when we talk about how these viroids cause disease. Okay, this is how they replicate. This is the PSTVd. These are the viroids that reproduce in the nucleus. So the viroid is shown here, the green circle of RNA. It gets into the cell. And again, you've seen how they get in cells, either through pollen or insects putting them in and so forth. And they go to the nucleus. There they're copied by Pol II into concatemers. And so here are 2 unit lengths or 2 genome lengths, but they're longer than that. Just make it clear we made it short. And then the other strand is made, copied to the other strand. And interestingly, this one gets into the nucleolus, and there is the enzyme that cleaves the concatemers into unit length molecules. And those are circularized, again, by host enzymes. They come out of the nucleolus, they come out of the nucleus into the cytoplasm, and then they move to the next cell. Plant cells are different from ours. They have these plasmodesmata that connect the cells, and this is how it moves from cell to cell, and eventually we'll get through the whole plant, of course. So that's the PSTViroidae, the ones that reproduce in the nucleus. Then we have the avsunviroidae, which are the ones with the hammerhead ribozyme. These actually go to the chloroplast. So it's getting into the chloroplast. It is replicated in this case. Now, you wouldn't predict this, but this is what happens. There's a DNA polymerase in the chloroplast that copies this viroid RNA. It's copied to form concatemers. The concatemers are cleaved. They fold up into the correct structure there, and then they can be copied again to the genome strand, cleaved, and there you got your final ones there. Then they move out of the chloroplast and move to the next cell through the plasmodesmata. Okay, so that is quite interesting that they are using or being reproduced by enzymes in the cell, and it's an RNA molecule. All right, where did they come from? Well, they came from the RNA world, I'm quite sure. We discovered them in the 20th century, where probably they were in wild plants. And of course, we use wild plants to breed modern crops, and we transferred them essentially, introduced them into our agriculture, but they've been in wild plants for many, many years, for as long as there have been plants probably and even before. And part of the problem is that crops globally are monocultures. They're all using pretty much the same seeds, provided by the same companies, and that's not good, because they get susceptible to diseases like viroid-induced diseases. Wild plants are really resistant to many diseases because they're genetically outbred, but our crops are not, so that's the price we have to pay. And as I said, they're spread now among crops by mechanical transmission, by contaminated farm machinery or equipment, even hands of the farmers, from plant to plant, pollen, bees, other insects, and other vectors as well. Now, finally, how do they cause disease? So the cool part here is that the genome gets into a plant cell, and the plant has RNA-based immunity, right? It sees an incoming RNA, and it chops it up to make small interfering RNAs. Some of those actually target plant genes by chance and silence them, and that's how you cause disease. So again, the viroid's chopped up on the way in as part of the plant defense, but some of the small RNAs that result from the chopping up can silence plant genes and cause the symptoms of the disease. Makes perfect sense, right? Because there's no protein encoded in this. So it makes perfect sense that this is an RNA-based pathogenesis. And experiments have been done to show that symptoms in the plant correlate with the production of small RNAs. You can take this pathogenesis region, which is where the small interfering RNAs originate, and show that these small interfering RNAs that cause disease map to this region. So you can change them and show that the viroids don't make disease anymore. So I think that's pretty neat that plant is really, it's immunopathology, right? This is really immunopathology. All right, question 1. Which of the following is not a property of viroids? Some contain a ribozyme, replicated by host RNA polymerase, metastable capsid, circular rod-like genome, none of the above. Which is not a property of viroids? Which is not a property of viroids? Metastable capsid, yes. Of course, there's no capsid. Everything else is correct. Ribozyme, host polymerase, circular genome. Let's move on to satellites. This is not the kind that are orbiting the Earth. They're really viruses. So I'm going to make a case that we shouldn't call them satellites, but I'm going to start because historically that is what they were called. These are viruses that depend on a helper virus for propagation. We've sort of touched on this when we talked about retroviruses. Remember, Rouse's virus was not defective, but all the other transforming retroviruses were defective because they lost genes and they can't reproduce without a helper. It's the same thing here, except these are naturally occurring. And these satellites can be RNA, they can be single-stranded RNA, they can be DNA or circular RNA genomes. And what makes them dependent on a helper virus is that they lack genes needed for replication. And there are two kinds of satellites. There are satellite viruses, and these encode structural proteins that will actually encapsulate the genome. They make distinct particles from the helper virus. And then we have satellite RNAs, and these are packaged by helper virus proteins, and they also rely on the helper virus for replication. These may or may not encode protein. So here's an example of a satellite RNA is encapsulated in a capsid made by the helper virus, and the helper virus RNA is in it as well. So you have both satellite RNA and helper. So the distinction is that the viruses encode a capsid. That's what makes them a virus, and the others are just satellite RNAs. Okay? So there are a couple of different kinds of satellite RNAs. I mean, the important part is that they don't encode capsid proteins, but you know, there are longer ones, class 1, 220 to 1,500 bases of single-stranded RNA, and these can have one open reading frame encoding some kind of non-structural protein, right? They don't encode structural proteins. Class 2 are a bit shorter, and they don't encode any protein. And finally, class 3 are circular, shorter even, and again, don't encode protein. So these have to be reproduced, of course, in a helper virus infected cell. They don't encode any protein at all. But they're all defective in a way because they depend on the helper. So the helper allows them to reproduce. They get packaged in the helper particle, and they endure. And these are in plant and insect viruses, but they're also mammalian examples I'll show you in a moment. This on the right here is just a schematic about how these could replicate. And again, this is a helper virus infected cell. The satellites do not reproduce on their own. But for example, here we would have the satellite RNA entering the cell. It's typically circularized. It's reproduced by a rolling circle mechanism. Again, the polymerase for this is supplied by the helper virus. So this would be an RNA virus polymerase. You make concatemers. Some of these helper RNAs encode ribozymes like the viroids, and they cleave those. So here are some unit lengths, which could then circularize and go through the reproduction scheme again. This is just showing two different ways it could happen. The concatemer could be cleaved, or the concatemer could actually be replicated and then cleaved. So that's satellite RNAs. And here are some examples of satellite viruses. And there's actually one that infects vertebrates. It's adeno-associated virus. We've talked a little bit about adeno-associated virus, AAV. We'll talk more about it next time because it's a big virus used for gene therapy. The helper is either adenovirus or herpes virus. And that's why they're originally called adeno-associated, because adenovirus was first found to be the helper. These are viruses with single-stranded DNA. They make particles. The AAV encodes its capsid, 4,700 nucleotide genome. Here, the capsid protein, and the host's vertebrates. Many AAVs infect us and other animals as well. Then there's an insect one. The helper virus is chronic bee paralysis virus. The satellite is called chronic bee paralysis virus satellite, right? So that's the pair. And this satellite, single-stranded RNA, again, a particle, 1,100 nucleotide genome, RNA genome, 15 kilodalton capsid protein infects the bee, of course. And then finally, a plant example. Tobacco necrosis virus is the helper, and the TNV satellite is the satellite. The satellite encodes a capsid protein and infects plants. So there are many examples of these throughout. So here's the reproduction cycle of adeno-associated virus, just to show you that this is not unique to insects or plants. Here we have AAV infecting a cell. Remember, these have these weird single-stranded DNA genomes with the T structures at either end. We talked about how they reproduce ages ago. But this requires a helper adenovirus to go through the reproduction cycle. And in particular, E1A and some of the early adenovirus proteins are needed to get synthesis of mRNAs of AAV. And so you'll see next time that when we use AAV for gene therapy, we don't actually co-infect with adenovirus. We just take the genes, the adenovirus genes that are needed, we put them on a plasmid, and we put them in the same cell. In plants, they cause distinct disease symptoms that are typically not seen with the helper virus alone. So if we have a plant virus, the helper virus on its own can infect plants and may cause a certain kind of disease. But when the helper is present with, when the satellite is present with the helper, you can get a different kind of disease. And this is an example of chlorosis. You have parts of the leaf that have lost chlorophyll, so they're no longer green. You can also have necrosis, which would be destruction of the cells. And so they cause a distinct disease compared to the helper. And a key point here, satellites are not defective viruses derived from the helper. They have no homology with the helper whatsoever. They're entirely different viruses, and how they evolved and so forth is not clear. Now, there is a satellite virus of us, of humans. There's just one that we know of, and that's hepatitis delta virus. So I'm not calling it a satellite now. I think we should call these viruses, as long as they're in a particle anyway. And this is very interesting because it has properties of both a viroid and a satellite, and its helper virus is hepatitis B virus. We think that when you have both hepatitis delta and B virus together in your liver, it makes hepatitis worse. Some controversy over this, but I think it's correct. About 18 million people are infected globally with hepatitis delta virus. That's about 5% of the 350 million carriers of hepatitis B virus, right? That's a number we put forth when we talked about HBV. A lot of people are infected. About 5% of those have both. You cannot have hepatitis delta alone. It will not reproduce. You have to have them both. And hepatitis delta used to be very high in Europe, and it's gone way down there because of control of Hep B, but it is quite high in the Asia-Pacific region still, as you can see by this map, where the shade of the gray or black or white shows you a very high, intermediate, low prevalence of hepatitis delta. But basically, if you get rid of Hep B, you will get rid of Hep delta. The genome is shown here, very much like a viroid, right? It's a circular single-stranded RNA, extensively base paired. And this one is 1.7 kilobases. In an average liver cell, you'll find about 300,000 copies. So that's the genome. And then there's an antigenome that is made in infected cells. Of course, this one is the one that codes for proteins. And so the mRNA will be the same polarity as this antigenome. So it's almost like a negative strand RNA virus in a way. And this encodes an RNA, about 50,000 copies of this antigenome. And this has a ribozyme in it, and that's the red dot here, which is labeled self-cleavage. So that can cleave itself. And its relevance in reproduction will be evident in a moment. And then you make an mRNA from this, which is the same polarity as the antigenome. And it encodes the single protein made by this, or encoded in this genome, which is delta antigen. And there are two different delta antigens. They're the small and the large delta. Small is made by translation of the open reading frame shown here in orange. But RNA editing occurs on a subfraction of these mRNAs and removes the stop codon so that you get a slightly longer protein. So RNA editing is a process where bases are changed without being templated. You can have, for example, insertion of bases or deletion of bases during polymerization. And so that's how you get the second protein. So again, the smallest is the S, and then the editing gives you a larger version of that protein. And what does this do? What does the delta antigen do? Well, it helps it to get packaged in the hepatitis B virus coat. Now remember, hepatitis B virus, which is the helper for hepatitis delta, it is an enveloped virus, and it's a DNA virus. So remember, the DNA is encased in an icosahedral capsid. It's the funny DNA that's gapped and has a protein and an RNA on it. This is encased in an envelope, and the envelope has the hepatitis B virus spike proteins in it. And there are different kinds. There are large, medium, and small spike proteins here. But it is the spike in the envelope. Well, of course, the spike drives the formation of the envelope, right? And so the hepatitis delta genome, which is shown in here, is encapsulated in this envelope with the hepatitis B spike proteins in it. And those spikes, of course, bind to the same receptor as Hep B binds to. Now, the delta antigens are shown here as well. The large delta antigen is a membrane-associated protein, as you can see there. And we believe that it helps to package the RNA genome here. And the small delta antigen coats the RNA genome. So that's where the two delta proteins go. And the envelope and the glycoprotein, again, comes from hepatitis B virus. So that's what B provides. Hepatitis B virus provides encapsulation. The rest of the process, the reproduction, is done by the host. And again, by the host RNA polymerase, very much like we've talked about for viroids and satellites. So here's a schematic of the reproduction. You start with the genome, which is the not minus strand, basically. It gets into a cell, goes in the nucleus, and it's recognized by Pol2. And Pol2 will copy it and make a plus strand. And it makes concatemers, right? Rolling circle concatemers. And the red is the ribozyme. And so self-cleavage can give you essentially what is the mRNA encoding for delta antigen, as I showed you. You know, that requires a polyadenylation, so it gets further processed after the ribozyme cleavage. But some cases, only the ribozyme cleavage occurs, and that gives you the circular plus-stranded genomes, full length. And then those are copied again by the same RNA Pol2 to make the minus strand. Again, concatemer, which are resolved by ribozyme cleavage. So, you know, resolution simply means making unit lengths from the concatemer. And that is what is going to be packaged, of course, into the hepatitis B virus envelope and spike glycoprotein. Now, we used to think that this was unique to people, this hepatitis delta virus, but just in the past couple of years, delta-like viruses have been found in other animals, birds and snakes in particular. And this came about by sequencing RNA transcripts. And there are two separate reports. One used healthy birds and the other used sick snakes. They found hepatitis delta-like sequences in both circular RNAs, very much like hepatitis delta virus. Kicker is there's no hepatitis B viruses in these birds and snakes to act as a helper. Yet, the only thing encoded in these genomes is delta antigens, so there has to be a helper somewhere. By the way, on the left is a phylogenetic tree showing you the relationships between these delta viruses. So on the top are human isolates. They fall into two branches on this tree, and there's a lot of them, as you can see by all the different branches. And then down here are the snake and the bird HDV-like sequences. So they're quite evolutionarily different, although they're similar to each other. Now, since this work was published, it looks like this is a bona fide virus of birds and snakes. It looks like even they can be spread among snakes in colonies. Other people have found that other viruses can actually be helpers for hepatitis delta in cell culture. So you can take the delta RNA and put it in a cell and then infect with another virus. You can infect with hepatitis C virus, vesicular stomatitis virus, dengue virus, West Nile virus, and they will all package the delta virus genome. Now, we don't see this in people. It's never seen. The only helper in people is hepatitis B virus. But in the lab, it shows that in principle, other viruses can package the delta genome. And so maybe in snakes, some other virus is being the helper. They didn't find any other virus in these transcripts, but then again, it may not be a virus that we know of, so you wouldn't recognize it. So stay tuned. And this makes sense, because why should delta viruses be unique to humans, right? You should be able to find them in other animals. Just a matter of looking. If you haven't found something, it's probably because you haven't looked. Which of the following statements about satellites is correct? There are no human satellites. They are defective viruses derived from the helper. Like viroids, they do not encode protein. In plants, they cause symptoms distinct from helper virus. All of the above are correct. All right, so here we have a bunch of, and in plants, they cause symptom distinct from the helper virus. That's correct. There are no human satellites. Well, that delta satellite was originally called a satellite. They are defective viruses derived from the helper. I said they're not. They do not encode protein, but they do. Satellites can encode protein. To end this part, I want to talk about virophages. This is a term given to us by the French, who are, as I've said before, very good at making up interesting names, like Mimi virus and Mama virus and Moo virus. So they made up this name virophage, which is derived from phagein. You know, bacteriophage comes from the Greek meaning to eat. And so they call this virophages, which means eater of viruses, right? Virus eater, which I totally disagree with. But that's what they called it, and it seems to be sticking. You know, it's sticky. These are circular double-stranded DNA viruses. They have icosahedral capsids. And again, they're helper dependent, but these will replicate only in cells infected with a giant virus. And they seem to interfere with helper virus replication. Here are some virophages that have been discovered. The first one, called Sputnik, it's a satellite, right? So you may remember Mimi virus, the first giant virus discovered, found in a cooling tower in Paris. And so was the Sputnik, which uses Mimi virus as the helper. So that's the size of the Sputnik genome in the open reading frames. And then here are some other virophages. Ma virus from Texas, coastal waters. And this is its helper here, Cafeteria rombergensis virus. By the way, this organism is a marine phagotropic flagellate. It's an interesting little eukaryote that it's very numerous in the oceans, and it eats a lot of things, which is why they call it cafeteria. Anyway, there's a virus of cafeteria, virophage of cafeteria. Then there's organic lake virophage, Sputnik 2. This is funny. This was isolated from the contact lens fluid of a patient with keratitis in France. And the helper virus is called lentille virus. Lens virus in French. Isn't that cool? And then a bunch from Yellowstone Lake, lake in Antarctica. But this map here shows you can actually take the ocean data sets. You know that Craig Venter went around the world in his yacht, and he sampled the ocean and sequenced it, and you can access all of that sequence information, and you can find virophage sequences in the data set. You can see they're all over the world. In the oceans, they're also on land as well. So virophages are everywhere. Here's an example of what they do. This on the right is a picture of the Sputnik virophage that infects Mimi virus infected cells. Right? These virophages cannot reproduce on their own. They have to be in a cell infected with another virus, and that's why they're, again, helper dependent. Let's see. Here's a mimivirus, top left, panel A. These bigger guys are mimiviruses, and they have the typical fibers on the particle. And this cell is co-infected with Sputnik, and Sputnik are the smaller ones right there, the smaller particles, which the reconstruction of it is shown at the right. I mean, they're respectable particles. They have a lot of capsid proteins. So mimivirus is the helper. Sputnik is the virophage. And again, the Sputnik will interfere with the yield of mimivirus. That's why it was called virophage. It doesn't actually eat mimivirus. It just interferes with its reproduction. It lowers the virus titers, makes weird particles. So this is a mimivirus which has Sputnik in it. So there's no mimivirus genome, so that particle is going to be defective. And then down here you see a mimivirus particle that's all morphologically aberrant. Here's some more on the left here. So this is all done by Sputnik. Sputnik really messes up the ability of mimivirus to reproduce. So they steal from mimivirus, and they also make aberrant particles. And this is typical of all of the virophages. Oh, here's a picture of Cafeteria rowenbergensis. Isn't it cute? You know, it's a little single-celled eukaryote. The ocean's full of them. It's a marine phagotropic fly. It eats things, hence the name. So there's a virus of Cafeteria. It's called C. rowenbergensis virus. And then Ma virus is a virophage of Cafeteria. It will interfere with its yield. Then there's the organic lake virophage, viruses that infect algae, Phycodnaviruses infect algae. And as I said, these interfere with the yield. When you infect when you infect amoeba with mimivirus, the Sputnik virophage will reduce yields by 70%. You also inhibit lysis. Now, this organic lake virophage, organic lake is a hypersaline meromictic lake in Antarctica. You got to look that up. That's a cool word. But it's in Antarctica. It's a hypersaline lake there. There's a virophage of Phycodnaviruses. So when this was discovered, the authors speculated that this virophage can influence the composition of the microbial community in this lake. And so they modeled the effect of virophage on the microbes that are infected with Phycodnaviruses. And without the virophage, there's cycles of virus reproduction, death, and cell growth or blooms. The effect of adding the virophage to the equation is the frequency of the blooms increases. And this suggests that the virophage reduces the mortality of the algae caused by Phycodnaviruses. Now, these lakes in Antarctica have long cycles of daylight and darkness, and so a decrease in Phycodnavirus cell killing may be essential for maintaining the stability of the microbial food web. So that's a cool idea of what these might be doing. They probably also exchange genes. You can find genes in them that come from the helper virus. And I've just told you that they may even have an impact on the ocean ecology. So they're very cool and remain to be studied even more. Are they satellites? So when they were first discovered, people started calling them satellite. And the people who worked on Sputnik, et cetera, got offended. I was at meetings where people had these big arguments saying they're not satellites. So I think they're not satellites, they're viruses. Like many autonomous viruses, they depend on the transcriptional machinery, except in their case, it's from another virus, not a host cell. Many viruses are dependent on transcriptional machinery of the host. They're all dependent on translational. We don't call them satellites, we just call them viruses. This happens to be provided by another virus, so it makes perfect sense to me to call them viruses and just point out that they depend on a function from another virus. That brings us to the last topic for today, which is prions. Infectious proteins with no nucleic acid. Now, these are periodically in the news. BSE, bovine spongiform encephalopathy, aka mad cow disease, you've heard of, Creutzfeldt-Jakob, scrapie, kuru, chronic wasting disease of deer and elk. These are periodically in the news because the news likes to report things that are going to wipe out humanity, of course. 1997 Nobel Prize in Medicine went to Stanley Prusiner for really developing the theory of the prion and getting the experimental data. And nobody believed him at first. I read his autobiography a couple of years ago. It's fabulous. He just talks of how nobody believed him for years. They would yell at him at meetings, telling him he was wrong. And he turned out to be right, and he got a prize. I love this sign. Wouldn't you love to live on Prion Road? It is not a through road, as you can see. Here's an article from the New York Times. In 1984, George Balanchine died of Creutzfeldt-Jakob disease, a very famous ballet dancer and, of course, a choreographer later in his career, New York City ballet head. And at one point, he couldn't walk anymore and slowly lost all his function and died. And he had Creutzfeldt-Jakob, a prion disease. All right, so what is this? This is a transmissible spongiform encephalopathy, TSEs. And encephalopathy is a disease of the brain, simply. Remember, encephalitis is an inflammation of the brain. Encephalopathy is just a disease. And these TSEs are fatal neurodegenerative disorders of mammals. Mammals only, as far as we know. Several thousand humans are diagnosed every year with TSEs in the whole world, as far as we know. And about 1% of those actually arise by infection. The others are sporadic or genetic, as you'll see. By 2002, 120 humans had contracted one of these TSEs, Creutzfeldt-Jakob, from eating meat from animals with BSE. Key here is that I don't care how well done you like your burger, you cannot kill a prion. It resists autoclaving. So think about that next time you have a burger. There are many TSEs of non-humans, and these are the names of them. There's BSE, or mad cow disease, as it was called, bovine spongiform encephalopathy. We have chronic wasting disease, deer, elk, and moose. We have exotic ungulate encephalopathy. This is an ungulate here, Nyala and greater kudu. We have cats, feline spongiform encephalopathy. Your kitty at home can get it, and cats in the zoo, lions and tigers can get it. Scrapie in sheep and goats. There's even one of mink, transmissible mink encephalopathy. Cost of being a mammal, I suppose. And in humans, we have a number of distinct TSEs. They're all prion-based. We have Creutzfeldt-Jakob disease, CJD. We have fatal familial insomnia, Gerstmann-Straussler syndrome. We have Kuru, and then we have variant CJ, which you get from eating BSE-contaminated beef. It's different from CJ. Now, all of these have slightly different pathologies and incubation times and outcomes. That's why they have different names. Now, what does spongiform mean? So transmissible spongiform encephalopathy. The spongiform part is because your brain looks like a sponge at the end stage of disease. These are sections from people who have died, obviously. They have big holes in them. And so that's a problem, right? Depending on the cell type, that has gone away. And these patients can have very severe psychomotor dysfunction. They often stop talking towards the end of their disease. They don't move. Again, in those different diseases in humans, depending on which one you have, the symptoms are different, and each one depends on which part of the brain is damaged. So each of those diseases I mentioned has a characteristic symptomatology and pathology. The first TSE recognized was scrapie, and it was called scrapie because the sheep would scrape themselves or rub themselves on fences, and they would rub off their hair, as you can see here. The sheep would also develop motor disturbances, uncontrolled trembling. In Canada, it was called tremblement du mouton. In France, they get paralyzed, they have a weight loss and die within 4 to 6 weeks. And of course, this is not good if you're a sheep farmer, but it's been recognized as a disease of European sheep for over 250 years. You can find it in the literature. And in fact, it is endemic still in some countries. In the UK, about 1% of all the sheep every year come down with scrapie. And as you will see, this is the origin of mad cow disease, because at some point someone thought it would be a good idea to feed cows ground up sheep. You feed a cow meat, it grows faster as opposed to grass or corn. All right, so that's the first TSE that we recognized. The farmers were very smart, actually. They recognized that infected animals could actually transmit the disease to healthy herds, because they would give a sheep to another farm, and then that sheep would transmit scrapie to the other farm. So this suggested that there was an infectious agent involved. In 1939, they found that if you took a sheep brain from a sheep who died of this scrapie and ground it up and filtered it, the infectivity, and then injected into another sheep's brain. The farmers weren't doing this. These were scientists. the infectivity passed through a filter, right? So they said, man, maybe it's a virus. However, it turned out it was highly resistant to ultraviolet light, ionizing radiation, formaldehyde, all sorts of activities or treatments that inactivate viruses. And so the idea arose that there's no nucleic acid in this agent, and that's where Stan Prusiner ran afoul of the field. They said, he's crazy. How could there be no nucleic acid? There must be a little bit of RNA or DNA wrapped up in the protein that we just can't find. And he spent years proving them wrong. So here's an inactivation curve of the scrapie agent. So basically what you do here, you irradiate the agent, and then you do a biological assay. For scrapie, you inject it into an animal. You can inject it into hamsters, for example, and they will develop the disease. And then you do an inactivation curve, and you see how much radiation you need to inactivate infectivity. And these two lines are curves for RNA viruses and various DNA viruses. And you can see that DNA viruses with big genomes, you don't need a lot of radiation to inactivate them because there's a big target. The genome is big. And as the genome gets smaller and smaller, you need more radiation to inactivate the viral genome. Same with RNA viruses. And scrapie was off the chart, basically. It needed more irradiation than anything we knew of, which, to many people said there's no nucleic acid here. At some point, people realized that the animal TSCs that had been known and the human ones had similar properties, and they thought, we can study we could study scrapie as a model for human disease. And these include defects in plasma membrane formation, vacuolization of neurons, astrocytes, oligodendrocytes in the brain, loss of neurons, spongiform appearance, accumulation of glial fibrillary acidic protein in clumps in the brain, and amyloidosis, fibrils of misfolded prion protein. These are all characteristic of human and animal TSCs. And so the animals were used as a model to understand the pathogenesis of these diseases. And so eventually, as I said, we could make brain homogenates and inject them into susceptible species. We found that certain animals could be infected, like hamsters, and would develop a disease. Initially, these sorts of symptoms took many years to develop in the animal models, but they got better at doing that. In people, this is a long-term disease. Ataxia, dementia, and death happens after many, many years of incubation. Prions appears to accumulate in lympho-reticular and secretory organs, and it spreads to the CNS. And there, of course, the typical pathology of spongiform encephalopathy, loss of neurons, and so forth. Now, what's interesting, there's no inflammatory response. There's no antibody response. There's no T-cell response. It is not a foreign protein. It is your protein that is doing this, as you will see. You can never find in people with prion diseases, CJ, for example, they don't make antibodies to the protein because it's their protein. Now, these TSCs are very serious diseases. You can be incubating for years and you don't know it until you have symptoms. And then it's too late to do anything. And in fact, they are all untreatable. We have no way to alleviate symptoms. We have no drugs. People have been trying to make drugs for years, but there are none so far. 100% fatal. So thousands of people a year develop TSCs and they die inevitably. So in 1967, a mathematician by the name of Griffith actually suggested that the agents of TSC were protein. He was the first one to do that. And Prusiner took that up. And in 1981, he was able to purify infectious protein complex in the brain from scrapie sheep. He purified the protein, and he was able to transmit it to animals using the purified protein. And he called this protein a prion. It's a portmanteau of proteinaceous and infectious. Technically, this is pro-in, right? And in his autobiography, he said he was in the cafeteria at UCSF one day. He was eating a tuna fish sandwich. I don't know why we need to know that. But he said, I should make it pro-in. But he said pro-in didn't sound good. So he reversed the letters and came up with prion. And that sounds good. That just rolls off the tongue, right? So this is the protein that causes the disease. It is encoded by a gene that's been called the Prion gene, PRNP, and it's essential for the pathogenesis of these TSCs. So what we understand now is that the pathogenic prion is a conformational isoform of a normal host protein. So the normal protein is called prion protein C for cellular. This protein is on the outer surface of neurons. It's put there by a GPI anchor. We don't know what it's doing, but the abnormal conformer, when introduced in the organism, causes the PRPC to convert to the pathogenic conformation, which is called PRPSC, in honor of Scrapie, even though Scrapie has nothing to do with this anymore, right? Or ever did. It's just because that's where we discovered this abnormal conformation. So you all have a protein in you called PRPC, and very rarely it may get converted to PRPSC, and then you develop a TSC. Now, Prusiner originally found that proteinase K would completely digest the PRPC, the normal protein, in vitro. He used this as an assay, whereas the PRPSC, when digested with the same protease, proteinase K, would make, it was mostly proteinase resistant. You got a 27 to 30 kilodalton fragment, and he could use this as an assay to see if his animals were developing the PRPSC. And what we think happens is that the PRPC protein exists in this form. The structure has actually been solved, and the conversion to the pathogenic form involves the creation of many beta sheets, as shown here. And this has been validated by structural studies. Key, in my view, was the identification of the protein and then finding the gene in mice and knocking it out. So mice lacking both copies of PRNP, the prion gene, are resistant to infection. You need that gene there to be susceptible because you have to convert your cellular prion to the pathogenic form. You can introduce PRPSC into animals, into people by infection. This is what we call infection. You can eat contaminated food. You can get transplants of human products, and you can acquire the PRPSC, and then that will cause your PRPC to transform into PRPSC. Some of us have inherited mutations in the gene that predispose us to misfolding. And then whether you're infected or you have a familiar disease, the SC eventually accumulates, and that's what leads to the symptoms of the TSC. There are three types of these TSCs. They're the infectious or transmissible type. That's where you eat a burger contaminated with prions. Prion SC converts your PRPC into more PRPSC, and you eventually develop the disease. There's familiar forms where you inherited mutations in the PRNP gene, and we know exactly which kinds of protein changes will lead to spontaneous misfolding of your PRPC at some point in your life to give you, so you're predisposed to these diseases. And then we have sporadic, where there's no infectious part of it. You don't eat anything contaminated. You have no mutations in your PRNP gene. The protein just misfolds spontaneously, and you develop. Once you get a little bit of PRPSC, then it goes all the way. And all three of these diseases can be transmitted experimentally. We can inoculate animals with the ground-up brains, or we can feed them to them as well. There are a couple of different kinds of human infectious TSCs. There's Kuru, which I'll tell you about in a moment. There's infection of people by the transplantation of corneas, hormones, or transfusion of blood with patients who had CJ. We didn't know it because they had no symptoms. Cornea transplants, production of hormones from humans, all of this have been shown to infect people with prion diseases. And BSC arose in cattle by feeding sheep to the cows. And then when we ate the BSC contaminated beef, we developed variant CJD. Kuru was first observed in the 4 people of New Guinea. It is a disease with a long incubation period and results in a TSC. The people lose motor function, cognitive function, and die eventually. And Carlton Gajdusek, an American virologist, went to study it, and he found that it mainly occurred in women and children because of ritual cannibalism of the brains of deceased relatives. When someone died in the tribe, the brain was fed to the women and children as part of their ceremony. And so they got Kuru. And so Gajdusek and others convinced them to stop doing this, and it got rid of Kuru. So they were eating the brains of people with Kuru, And that's how they were infected. So somehow the prion goes from your intestine to the brain. We don't actually know how that happens. Then there's sporadic Creutzfeldt-Jakob, where the protein misfolds spontaneously. This is very rare. One or two out of a million humans globally. These people are typically 50 to 70 years of age. And this accounts for 65% of the thousands of cases of TSCs every year. It appears with no warning. There's no epidemiological indications. These patients have normal genes, and they can transmit the disease to others. If they donate blood or a cornea or whatever, they can transmit the infection. And we think Kuru became established in New Guinea when that first person developed sporadic CJD years ago. He died, and they started eating the brains, and that started the chain of infection. It became an infectious form after that. The familial encephalopathy is an inherited disease. There's a single autosomal dominant mutation in the gene, and these people develop it, and then they donate corneas, blood products, etc. They can transmit the infection to other people as well. So it arises in you sporadically, and then you transmit it. The CDC keeps track of CJD deaths. This is a graph of total deaths. The bars are total deaths from 1979 to 2015. So, 500 or so total deaths. And this is the rate for 1 million persons here in red. So slightly over, one per million people per year in the U.S. So it's rare, but as I said, it's 100% fatal. Here again are the three types: infectious, genetic, and sporadic. The genetic and sporadic arise within you, but the infectious, you ingest something. Here's a model for how maybe the PRPSC that you ingest may convert PRPC in your gut to the pathogenic form, and at some point it has to get to the CNS. We don't know how that happens. If you get a corneal transplant, they put the altered protein right in you, so that initiates the chain of infection. Why don't we digest the PRPSC? It's resistant to digestion. That's the kicker. Remember the proteinase K experiment? It's resistant. Now, BSE arose in the UK. It's a form of forced cannibalism because they were feeding cows ground up sheep. And some of the sheep had died of scrapie. So this is a protein supplement, so your cows grow faster. You grind up other animals, including sheep and pigs and who knows what else. It is fed to cows. And in 1970s, they changed the method for preparing it, which allowed the scrapie proteins to survive and pass into the cows. The previous method got rid of the scrapie protein, but was more expensive, so they decided to save money. The result was this BSE epidemic. And we stopped this byproduct feeding, and the outbreak stopped. But unfortunately, a number of people consumed beef and acquired this variant CJ by eating it. And here's the outbreak in cows and in humans. So the orange is BSE starting in about 1988. The number of cases increased. We estimate that a total of 1 to 2 million cattle were infected with prions, and they were mainly slaughtered to stop this. And then we stopped feeding cows, ground up animals. But then the human outbreak of variant CJ began because there's a long incubation period. You can see it peaked in about 2000 and then went down. And then there were a few cases of these patients transmitting it to others by giving blood. Those are the green bars right here. So it's variant Creutzfeldt arising from consumption of beef, and then variant transmitted by blood transfusions. Now, the problem here, of course, is that in cows, the incubation time for BSE is 5 years. We kill cows after 2 or 3 years for meat. They're ready to go. So you never know if they're infected or not. And that's why this entered the food supply. We still, to this day, have cases of BSE in cattle all around the world where cattle are grown. These are probably sporadic. Since we've modified the feed procedures, we're not feeding the cows scrapie anymore. So these are cows that sporadically misfold their prion proteins. And so we're very concerned about the food supply, but we don't test very many cows, right? We test less than 2% of slaughtered cattle. Yes, if you have lived in Europe, you can't donate blood. I know one of the co-authors of the textbook, Theodora, is from Greece, and she lived in the UK for a while, and she cannot donate blood because of this. Now we have diagnostic tests, which are good for screening cows, and we are looking for drugs to help treat both cows and people, but we don't have any yet. So these are the number of BSE cases. This is in cows now in North America, Canada, and the U.S. from 1993 to 2018. And U.S. is orange, Canada is blue. You can see some years there are more than others, 26 in all. Now you may say, oh, that's nothing. But if it gets into the food supply, it's not nothing. We're going to have another outbreak. So you have to be really careful here. And again, it's not about feeding sheep. It's about sporadic BSE arising in cows and making sure that doesn't get into the food chain before you know it. Very interesting case reports in the literature. I've written about 2 of them on the blog. One is a case of prion disease 15 years after this gentleman ate a burger in London, and he lived somewhere in the Middle East, I think, and he developed a fatal disease, which turned out to be a TSE that he acquired. It was variant CJ. And then this one is very scary. A guy came into an emergency room and shot himself in the head, and his brains splattered all over the ER. Well, turned out he had a TSE, and they had to throw out all the equipment in the ER, hundreds of thousands of dollars of medical equipment because you can't clean it. You cannot sterilize it. They have procedures for this now to try and get around this. So we've had 3 epidemics of CJ in humans. The first from injection of growth hormones purified from human pituitary glands, and those are from people with a TSE. We didn't know it. We started an outbreak of that, so we've stopped doing that. We make it recombinant now. Transplantation of dura mater grafts also transmitted CJ into people. And finally, variant CJ from prion contaminated meat. Beyond Burgers, yeah, I agree. I totally agree. Beyond Burgers are great. And the other company, there's Impossible Burgers, etc. Impossible Burgers were invented by Pat Brown, who's a Stanford professor, a microbiologist. Here's the assay for picking up prions. It's very cool. It's kind of like a PCR. You can take olfactory brushes, and now you can actually do urine and blood. You don't have to do this anymore. Take the sample, and what you basically do is a PCR, but it's with protein. So you take your sample, and if it has some PRPSC, you mix it with PRPC, and then PRPC will cause the conformation of the PRPC into longer monomers with incubation. And then you sonicate it to break it up, and then you go through the cycle again. And with time, you get more and more of these oligomers made of the transformed form of the protein, and you can detect those by an assay of various sorts. So you do cycles just like PCR, except you're allowing these multimers to grow. So now we can check urine and blood of people. We can check cows for them as well. So that's all good. The last thing I want to talk about is the species barrier. You can put disease brain material into the same species, and that's fine. But different species, it's inefficient. Mostly the sequences of the protein in the donor and the host and who you're giving it to should be the same. So the sequence of PRP is the host tropism determinant. So here's an example. If you take hamster prion SC from a hamster who had the TSC, you put it in a mouse, no disease. That's the species barrier. But if you make the mouse transgenic for hamster PRNP gene, then putting hamster PSC will give you disease. Okay, so the protein has to match with what you're injecting in. But some prions have broad host range. So if you put cow PRPSC into mice, you get disease. We have found that bovine PRPSC has a broad host range, and that's why we could eat cow meat and get a TSC from it. So that's why BSE is a concern, because the cow prion has a broad host range. Which of the following observations illustrate the prion species barrier? Mice lacking the PRNP gene do not develop disease. Mice fed hamster PRPSC do not develop disease unless they have a hamster PRNP transgene. Humans consuming BSE beef develop disease. Humans may sporadically develop prion disease. Humans may transmit prion disease to other humans via corneal transplant. Okay, that's right. Most of you got the right answer. B, mice fed hamster PRPSC do not develop disease unless they have a transgene. So mice lacking the gene do not develop disease. That has nothing to do with the tropism, the species barrier. Humans consuming beef develop disease. That doesn't illustrate the barrier. And the last two, they do not illustrate the barrier. This B does illustrate the barrier. The last thing I want to tell you about is a newly discovered disease, chronic wasting disease. which is a TSE of deer, elk, and moose, or cervids. And this is an animal with a TSE. Similar symptoms, similar disease development as we've been talking about. This is a prion disease. It's all throughout the U.S., as you can see here, and it's spreading. The numbers are getting higher every year. To 90% of deer and 60% in some herds are positive, and in wild animals as high as 15%. So both captive and wild animals are developing this disease. And deer shed the prions in their nasal secretions, saliva, their skin, blood. Giving birth sheds them, urine and feces. The carcass, which other animals can eat, is infectious, right? And so we're concerned about environmental passage of prions, other animals picking it up. We're also concerned about the prions getting into the environment. And then, of course, the deer will eat them again and get infected. So you have an ongoing cycle. And experiments have shown that you can take, say, an animal that's developed a TSE, grind up the brain, add it to the dirt. A plant will take it up. Prions in the leaves will be infectious. So you can take the leaves and grind them up and feed them to an animal, and it will be infectious. So there's some concern that these prions get in the soil, they get incorporated into plants, and other animals either contact them or eat the plant. So that's one of the reasons why we're worried. Now, so far, the host range of the cervid PRPSC is pretty tight. If you inoculate it into mice, no disease unless the mouse is transgenic for the cervid PRNP gene. If you make mice transgenic for the human PRNP gene, you infect then with PRPSC from deer, there's no disease. So this is good. It suggests that people are not going to be infected with this. But what about cows? You know, cows often occupy the same pastures as deer. Deer at night can come in and deposit their prions. If you put deer PRPSC IC into cows, you don't get disease. We're not sure if this is the ultimate word. So we're just worried if these deer prions would transmit to cows in contaminated pastures. Now, I know that you put it in the brain, you don't get disease, but the incidence is increasing over time. The number of cases are going up. And as you know, hunters like to shoot deer and eat them. So there's this website where they tell you to be careful. If the animal's acting weirdly, don't shoot it. You should wear gloves. You should not saw through the bone. You should avoid the brain and spinal cord. Wash your hands. Don't eat the brain, spinal cord, eyes, spleen, tonsils, lymph nodes. These are places where the prions tend to accumulate. You shouldn't eat meat from an animal that tests positive, and you can get your animal tested. But the concern is that, with more and more deer being infected, the number is going up. And of course, the protein can change. This is kind of a summary of TSEs, how they all arose. So maybe the earliest one was scrapie in sheep, which was then transmitted to deer and has started the chronic wasting disease outbreak. Or maybe the deer developed a sporadic infection that was then transmitted to deer. We really don't know. But the scrapie, of course, has led to mink having their disease because we fed sheep offal to mink. So now we have TSE in mink. And cows, of course. The cow outbreak infected humans, and we have passed it on to other people iatrogenically. And we also pass the prions of sporadic cases to others via transplants and blood donations and so forth, as I've told you. From the cattle outbreak, we have infected many other animals who are given cow feed. BSC has led to infections of other cattle. It has led to infections of animals in zoos and any animals that were fed cow meat. And even pet cats are often fed, cow meat, and some cats were infected with BSC beef, and that started the feline spongiform encephalopathy. So we have a naturally occurring disease and then others that are artificial. Next time, our last lecture, we'll talk about turning viruses into good things, therapeutic viruses.